Philippines is an archipelagic country famously known for having rich and diverse bodies of water. It is one of the reasons why tourists tend to flock to this country during the summer. But underneath this abundance lies something far more serious in nature, something that could cause a very contagious infection: Cryptosporidium parasites. Cryptosporidium species are protozoans that are known to cause waterborne infections, particularly diarrhea, in several parts of the world. Cryptosporidiosis, the infection caused by Cryptosporidium, is the second leading cause of infantile mortality, coming close to rotavirus infection. Any person, immunocompromised or not, may contract this parasite from waters contaminated with stool. According to the 2018 data of the World Health Organization, as many as 844 million people do not have access to drinking water service, with 159 million still dependent on untreated surface water from bodies of lakes, ponds, and streams. The worsening climate crisis, along with the growing population and urbanization, are just some of the challenges that further exacerbate this lack of access to safe drinking water. Southeast Asia, in particular, is highly susceptible to the health impacts of climate change and water crisis. As people continue to consume untreated water, the chance of contracting waterborne parasites remain high. At present, the Philippines still lacks sufficient literature on the epidemiology and management of Cryptosporidium species across the country. This is why Masangkay and colleagues aim to produce nationwide data on the microscopic and molecular prevalence of Cryptosporidium in the Philippines by using low-cost and sustainable methods. 23 study sites, all of which were large lakes and major water reservoirs, were established across Luzon, Visayas, and Mindanao, and were further subdivided into 29 sampling sites and 72 sampling points. Samples of surface water, bottom water, as well as substrate-associated biofilms were collected by the researchers for laboratory processing. In order to visualize the pathogens that might be present in the samples, different staining techniques were employed by the researchers in the lab. Staining samples helps determine certain biochemical and structural components which may aid in the subsequent identification of species. From the processed samples, 1 cm diameter smears were prepared and subsequently stained with modified quinones and modified safranin methylene blue stains, both of which are used for visualizing acid-fast parasites such as Cryptosporidium oocysts. For the fluorescence techniques, Auramine and direct antibody fluorescence technique were employed. Direct antibody fluorescence technique is a method that utilizes antibodies tagged with specific fluorescent dyes. This molecular conjugation reacts with the antigen of an infected cell in a sample, creating an antibody-antigen complex that fluoresces after a period of incubation. It can then be visualized using a microscope with a UV source. After staining, each sample smear was examined under 200 oil immersion fields and 200 high-power fields for traditional and fluorescence microscopy, respectively. Cryptosporidium oocysts were then compared with that of C. parvum positive controls based on measurements and other morphological characteristics. In addition to the low-volume water samples used for microscopic analysis, 1 liter bulk surface water from each sampling site was also collected for molecular analysis. Polymerase chain reaction was performed using a modified non-nested protocol in order to amplify specific DNA fragments from the samples. Gel electrophoresis was subsequently conducted in order to visualize the amplicons. Amplicons that were from PCR-positive samples were sent to Macrogen South Korea, a sequencing company, where they were further inspected before being deposited in GenBank. GenBank is a popular genetic sequence database that offers a collection of publicly available sequences submitted by different laboratories worldwide. By identifying these sequences, the researchers were able to perform a molecular evolutionary analysis of the Cryptosporidium species present in the Philippines and provide important data regarding their prevalence. Their results showed that the IFT method provided the highest positivity rate among all the other staining techniques employed, with 59%, 38%, and 50% detection obtained from the surface water, bottom water, and substrate-associated biofilm respectively. Modified quinones and safranin methylene blue might have been cost-effective stains, but they produced lower detection rates. From the stained samples, the Cryptosporidium oocysts that were detected had exhibited morphological features that were consistent with those of CDC's published descriptions of Cryptosporidium species, as well as those observed in the C. parvum positive control. In the MK and SMB stained samples, the oocysts appeared to be round, measuring 4 to 6 micrometers in diameter, and stained bright red against a blue background under 1,000 times magnification of light microscopy. The same morphological characteristics were observed in IFT. However, instead of red, they emitted bright green fluorescence against a black background in 400 times magnification. In terms of the theoretical approximates of oocyst counts, a range of 0 to 1 Cryptosporidium oocysts were detected in a 1 cm diameter smear region equal to 25 microliter suspension, which theoretically translates to 0 to 2,000 oocyst counts per 50 ml. According to Masankay and colleagues, this approximate result could already be considered a substantial contamination when compared to the lower estimates reported in Thailand, Australia, and Iran. Their careful preparation of sample suspensions might have minimized the loss of oocysts, especially when processed in multiple sequential steps. Additionally, the standardized 1 cm diameter smear that's coupled with systematic microscopy might have helped increase the detection of oocysts. However, the actual density of Cryptosporidium oocysts in some surface water samples was hypothesized to be lower than the theoretical value due to the uneven distribution of oocysts and presence of substrates in some environmental matrices. Thus, theoretical estimation shouldn't be considered absolute. All in all, 6 sampling sites tested positive with C. parvum, C. hominis, and other Cryptosporidium species based on the 1 liter bulk surface water samples collected. In Luzon, the prevalence of Cryptosporidium was reported to be 15%, equivalent to 2 out of 13 sampling sites. C. parvum was detected in Yambo Lake, and C. hominis was present in Ambuklao Water Reservoir. In Visayas, there was a 33% prevalence equivalent to 1 out of 3 sampling sites, namely in Bito Lake. In Mindanao, there's a 50% prevalence equivalent equivalent to 3 out of 6 sampling sites, C. parvum was detected in Pulangi water reservoir and C. hominis in both Lanao Lake and Tagunay River. The total molecular prevalence of Cryptosporidium in the Philippines was reported to be 21%, representing 6 out of the 29 sampling sites. C. parvum, C. hominis, and other Cryptosporidium species had an overall prevalence of 7% or 2 out of 29 sampling sites, 10% or 3 out of 29 sampling sites, and 3% or 1 out of 29 sampling sites respectively. In terms of the PCR results, the 18S or RNA PCR showed a high diagnostic sensitivity rate of 96.9% and specificity of 98.4% with an analytical sensitivity of 1 to 10 oocysts per action. The modified non-nested procedure employed has also saved a total of 5 to 6 hours of runtime per batch, although it did not have an effect on the sensitivity of the PCR. Masangkay and the other researchers had speculated that the microscopy positive but PCR negative samples might have been affected by the density and features of suspended particles, given that the study sites were considerably polluted. Currently, there is no policy in the Philippines about the detection of Cryptosporidium and other distinguished waterborne protozoan pathogens. This study provided the first national molecular epidemiologic data on the existence of C. parvum and C. hominis in the Philippines. The findings also supported the inclusion of Cryptosporidium and other waterborne protozoan pathogens in water quality assessment of freshwater resources and drinking water. Hence, the results of the study show that the Cryptosporidium oocysts were abundant in surface water, sediments, and substrate-associated biofilms from environmental freshwater systems. It is also detected that the use of low-volume Cryptosporidium with multiple matrices and sample area techniques are low-cost, user-friendly, sustainable, and reproducible methods. The use of standardized and systematic 18S or RNA PCR has been found to be a successful technique in detection of Cryptosporidium oocysts from low-volume environmental aquatic matrices. Since there is still no established practice to detect Cryptosporidium in aquatic environments, Cryptosporidium and other waterborne protozoan pathogens should be included in water quality assessment and monitoring according to the Climate and Health Framework.